So we've talked about glycolysis and about the citric acid cycle. So we have completely oxidized glucose now. We started off with our 6-carbon molecule of glucose, and it's gone. We broke it all down. We have exhaled all of the carbons as carbon dioxide. But we were able to make some really high energy molecules along the way, such as NADH and FADH2. So this next step about oxidative phosphorylation and the electron transport chain explains what happens to those NADH and FADH2 and how they give us so much energy. So we took all of the energy out and it's being stored in NADH. So remember that the breakdown of glucose happens really slowly because otherwise it would just be a huge release of energy and our cells wouldn't really be able to use it, just like how if we were to light an entire tank of gas on fire at once, our cars wouldn't go very far, right? We want to slow the release of energy. And NADH and FADH2 provide this connection between glycolysis and the citric acid cycle and oxidative phosphorylation. So they are that link. They take the energy from glycolysis and the citric acid cycle and they bring it directly to the electron transport chain. And that energy is now used to make more ATP, much more so than substrate level phosphorylation, which is what made the ATP during the citric acid cycle and during glycolysis. So, about the electron transport chain. It is a bunch of molecules that are embedded into the actual membrane of the mitochondria, so the inner folded cristae. It's each of them is a series of several molecules. Most of them are proteins. You don't need to know specifics about them, but you should know that the reason that the mitochondria is so folded like this is to provide extra space for more electron transport chains. Because if it was entirely flat and just like there's no picture of it. If it was just touching the inside of the outer membrane, it would have far less surface area and we could have far fewer electron transport chains. So since it is all inner folded and everything, it's just giving us more space to have more electron transport chains to make more ATP. So that's an example of structure fitting function. The structure of the mitochondria is a highly folded inner membrane, and that fits its function, which is that it needs the inner membrane to make ATP. So, most of these proteins exist in a multi-protein complex, which means a bunch of proteins working together. You don't need to know specifics about that. You maybe should be able to recognize that a cytochrome, it's bolded in a textbook, a cytochrome is one of the proteins that are found in the electron transport chain, but you don't need to know more than that. So basically, what happens is these proteins are in a line, and they just pass the electrons down. They start off at one end, pick up the electron, they pass it to another one. So the proteins are repeatedly getting reduced and then oxidized. So when they accept electrons, they are accepting something that's negative, they get reduced. They are reduced for a moment, and then they pass on the electrons to the next protein, and as they pass on the electrons, they become oxidized because they got rid of electrons. The next protein is accepting electrons, accepting something that's negative, it gets reduced. Then they pass on the proteins to the electrons to the next protein and become oxidized. So the proteins are reduced and oxidized over and over again. And this picture shows how the free energy of the electrons changes as they're passed on. So NADH drops off the electrons right here, and then they're passed all the way down through all of these different molecules. And so each time that they're passed, they lose some energy. So up here they have much more free energy than at the end. And that makes sense because they're losing potential energy every time they go down. And each protein is a little bit more electronegative than the previous one. And that's why they move in that direction. So the electronegative proteins are pulling the electrons towards them. And the most electronegative of the whole thing is this oxygen atom down here. So each protein picks up the electrons and passes it on to the next more electronegative protein. So of all the proteins in the whole electron transport chain, these are the least electronegative and these are the most electronegative. The most of the entire thing being the oxygen, which takes it at the end. And this is important to note because we'll talk about it later. NADH drops off proteins on the electron transport chain. other way around. No, NADH drops off electrons on the electron transport chain further up than FADH2 does. And we'll talk about why that is and what that means for ATP production. And so you can see here that the electron transport chain, this is the whole thing. We can see that we put in NADH, we put in FADH2, but there's no ATP made at the electron transport chain. Instead, what the electron transport chain does is it slowly lets the electrons lose their energy. So each time they drop down a step, they're slowly releasing energy. As you can see here, over here on the y-axis is the free energy. They're slowly losing energy rather than letting it all go at once. And what they're doing is building an electrochemical gradient. So let's see what that means. So what this says is electrons are transferred from NADH or FADH2 to the electron transport chain. You know that. Electrons are passed through a number of proteins, including cytochromes. You don't need to know that. Just know that there are some proteins called cytochromes. Just know what that word is. And the electron transport chain makes no ATP directly. So what actually does make the ATP is what we call chemiosmosis. So electrons transfer. So when the electrons are transferred through each protein down the electron transport chain, these proteins pump H+ across the mitochondrial membrane into the intermembrane space. So, is there a good picture of this anywhere? There it is. So, this is the start of the electron transport chain over here, and this is the end right here. So, this is water is down at the bottom right here. So, NADH drops off the electrons, and they're passed down through all the different proteins. Every time that one protein passes along the electrons, a proton is pumped from the mitochondrial matrix to the intermembrane space. So this is the space in between the outer membrane of the mitochondria and the highly folded cristae is the space right here. So these proteins are taking the energy that's released from the electrons. Because remember, each time they move down a step, they lose a little bit of energy. They're taking that energy and using it to pump a proton across this membrane. Protons are charged, H+, so they can't cross the phospholipid bilayer on their own, so instead they're picked up by this protein and moved across. It's an example of active transport. And the energy provided is from the electrons losing energy, and that's what it goes to do. So each of these steps pumps a proton across the mitochondrial membrane. So now we have an electrochemical gradient. We have way more H+ outside or in the intermembrane space than we do inside of the mitochondrial matrix. So, electron transfer to the electron transport chain causes proteins to pump H+ from the mitochondrial matrix to the intermembrane space. Now, we've created this super gradient. Basically, there's so many more H+ in the intermembrane space than there are inside of the matrix. And those protons, they do not want to be near each other. Just like if I were to make all of you students sit on one side of the classroom, you all would hate that, right? So these protons are looking for any way to get back across the membrane, and they can't cross on their own because they're charged. But there is a pathway that exists, and that's through a protein called ATP synthase. ATP synthase is a protein that exists in the mitochondrial membrane, and it allows protons to pass back through. This is ATP synthase. So you can see that there's way more protons out here in the intermembrane space than there is inside of the mitochondrial matrix. So these guys want to go from high concentration to low concentration. So what they do is they find ATP synthase, and ATP synthase has a channel that allows them to pass through. And as they're passing through, the movement of them moving, their kinetic energy of them moving, provides enough energy for ATP synthase to make an ATP molecule. There's a lot of mechanics happening here. The proton comes in, the rotor spins, blah, blah, blah. Basically, this proton enters the ATP synthase. It makes this rotor up here spin, and the spinning of that rotor provides enough energy for ADP to be joined to a phosphate to make ATP. So that is exactly how our ATP is made. So it's not made at the electron transport chain. It's made at a different protein in the mitochondrial membrane called ATP synthase. And this is called chemiosmosis because chemiosmosis is defined as the energy stored in the form of a hydrogen ion concentration gradient. So we have a hydrogen ion concentration gradient. There's way more hydrogen ions out here than there are in here, more in the intermembrane space than there are in the matrix. And so they use the energy stored there to drive cellular work, and in this case, the cellular work is making ATP. So this proton gradient, the fact that there's more protons out here than there are in here, is referred to as the proton motive force. So the proton motive force is what powers ATP synthase. So be familiar with that term, proton motive force. That's how powerful it is that there's more protons out here than there are in here. It can make all of this work happen. So chemiosmosis is found in, can occur in several different places, but here in the mitochondrial membrane, it does exactly what we just said, and the energy is used to make ATP. We'll see it happen in other places, such as in photosynthesis, and the energy is put to different work, but in this case, we're making ATP. So again, here's the electron transport chain. NADH and FADH2 drop off their electrons that they got from glycolysis and the citric acid cycle. They drop them off, the electrons move down through all these proteins, these protein complexes, and each time that they are passed down along, the protein accepts the electrons and they get reduced because they're accepting something that's negative. They give it up to the next one, so this first complex would accept the electrons. It gets reduced, then it passes them on and becomes oxidized as this guy becomes reduced. And each time that that happens, the proteins use this energy released to pump a proton across the membrane. So they are generating an electrochemical gradient of the protons. They are generating the proton motive force. Then the proton motive force is used through ATP synthase to make ATP from ADP. So we pumped all these protons out here into the intermembrane space, and then they find their way back through the membrane through ATP synthase. Okay, so let's talk about how much ATP is made at the through chemiosmosis, through ATP synthase. So for the most part, the energy goes glucose, NADH, electron transport chain, proton motive force, ATP. So you should know that. But these numbers are inexact. So if we look at this picture, you can see that it says 2 ATP are made in glycolysis. You knew that. Overall, 2 ATP are made. 4 are made, but 2 are spent, so overall 2 are made. 2 are made in the citric acid cycle, but about 26 or 28 are made in chemiosmosis. So why is that a question mark? There's a couple of reasons, but the main reason that you need to know is because NADH drops off their electrons up here, whereas FADH2 drops it off lower in the chain. So NADH allows for more protons to be pumped across than FADH2. So we have put more energy into the proton motive force through the use of NADH than we have through the use of FADH2. So it allows more ATP to be made because we pumped so many more protons across the membrane. This whole process is only about 34% efficient. So of all the energy stored in glucose, we have now stored 34% of it into ATP. Now, that might seem really inefficient. However, the most efficient cars that are on the road right now are only about 25% efficient. So this is much more efficient than any hybrid car. And the rest of the energy is lost to the universe as heat. And that is how we make 32 ATP through cellular respiration.